This podcast will deal with cardiac muscle. As you know, cardiac muscle is striated muscle, but it's involuntary muscle. The myofibrils in cardiac muscle are arranged quite similar to skeletal muscle, so much of what we talked about for how skeletal muscle contracts relates to how cardiac muscle contracts. Obviously, cardiac muscle is found in the heart and the proximal regions of the pulmonary veins. Now, unlike skeletal muscle, which the cells have multiple peripheral nuclei, cardiac muscles cells have central nuclei, usually one nucleus per muscle cell in cardiac muscle, but sometimes two nuclei. Cardiac muscle cells have a spontaneous ability to contract. We talk about Purkinje fibers. These are modified cardiac myocytes that play a role in impulse conduction, and there's a specialized junctional complex in cardiac muscle cells that are called intercalated disks that play a role not only in holding the cardiac muscle cells together, but play a role in cell-to-cell communication to aid in the coordinated contraction of cardiac muscle cells. This is just a histological view of cardiac muscle cells. I want to point out some key things about cardiac muscle cells. The myocytes are branched. You don't have to really remember the dimensions of them, but they're branched cells. They have intercalated disks, which are highlighted here by the arrowheads. These are the intercalated disks. The sarcomere contraction is pretty identical to skeletal muscle. The T-tubules are 2 to 3 times the diameter of the T-tubules in skeletal muscle cells, and in cardiac muscle, the T-tubules form dyads near the Z line. That's different because in skeletal muscle, the T-tubules are forming triads at the junction between the A and the I bands. The sarcoplasmic reticulum in cardiac muscle does not form terminal cisternae. Rather, calcium comes from the extracellular region through the T-tubules. The external lamina coating the T-tubules is where calcium tends to be stored for cardiac muscle. There's a tremendous number and volume of mitochondria in cardiac muscle cells. Mitochondria make up almost half of the cell volume in cardiac muscle cells, and that might compare to about 2% of the volume that mitochondria contribute to in skeletal muscle cells. So you might imagine, obviously, cardiac muscle, because of the high metabolic demands, have abundant mitochondria. They also have abundant sources of glycogen and triglycerides for energy. And cardiac muscle has abundant myoglobin because of the very high oxygen demand. There are hormone-like substances produced by cardiac muscle cells. One of them, called atrial natriuretic peptide, this is the marker for cardiac muscle. Atrial natriuretic peptide, we'll come back to this when we study the kidney, but it lowers the ability of renal tubules to conserve water and salt. But atrial natriuretic peptide and a related peptide, brain natriuretic peptide, found in ventricular myocytes of cardiac muscle cells, are markers for cardiac muscle cells. What I've shown in red would be the contrast points between cardiac muscle and skeletal muscle. So you can focus on these as you consolidate your studying between cardiac muscle and skeletal muscle. Focus on the red statements to look at the differences between cardiac and skeletal muscle. This is a typical histological view of cardiac muscle. Here you can see some cardiac muscle cells shown in cross-section, some shown in longitudinal section on the top of the slide, and in the middle one can see some of these specialized cardiac myocytes, the Purkinje fibers, important for impulse conduction and contraction of cardiac muscle cells. And we'll talk about the Purkinje fibers as we introduce them to you in the laboratory. This is showing the intercalated disk structure of cardiac muscle cells. It's the specialized junctional complex, and there is a transverse region to the intercalated disk that has desmosome-like structures and fascia adherens. That's on the transverse region. And then on the lateral region, there are gap junctions. So on the cartoon on the top, this would be the region of the intercalated disk that helps to hold the cells together. So here and here. And on the lateral region where the gap junctions are, this is how the cells are electrically coupled with each other. And notice in this cartoon, the intercalated disks are in the areas where the cardiac myocytes are branched. So as the branches come together and the cells stack together, that's where you have the intercalated disks, and they happen to be modifications on the Z line, because as you look at the structure, here's the A band, here's an I band, here's an I band, and here's an intercalated disk, a modification of this structure at the Z disk. If you were to trace the sarcomere, here's a sarcomere running from I band, the Z line on this I band, to the Z line on that I band. So fairly similar to skeletal muscle, but the intercalated disk, this tremendous modification at the Z line. Here's a higher power view of electron micrograph cardiac muscle. Here is the intercalated disk, this modification at the Z line. Notice here's part of an A band, part of an A band. You can see the lighter I band, and you would imagine the Z line running between, but this is the modification into the intercalated disk. On the lower power, here is the A band, the I band, like so, with the Z line, and then just the higher power view to see the intercalated disk. This is nice at the lower power. Here is the T-tubule system and the single cisternae of the smooth ER, so forming a dyad at the Z line. Here's a higher power view of the T-tubule with the dilated portion of the sarcoplasmic reticulum, so the dyad near the Z line. Clinical correlations for cardiac muscle. Cardiac hypertrophy, the myocytes become larger, but their number remains constant. Now, generally, cardiac myocytes do not regenerate. So if you have a heart attack, for example, a myocardial infarct, the dead myocytes are generally replaced by fibrous connective tissue. Now, there is some evidence from transplanted hearts that suggests that there's a very low percentage, perhaps less than 0.1% of cardiac muscle cells may have some ability to regenerate. And that's actually an area that people are very interested in, because wouldn't that be wonderful to be able to allow cardiac muscle cells to regenerate? There have even been experiments where people try to transplant satellite cells from skeletal muscle into diseased hearts, like of rodents, and try to see if those satellite cells can regenerate and transform into cardiac myocytes. And that research shows some promise. There's lack of calcium in the extracellular compartment leads to fairly quick cessation of cardiac muscle contraction in less than a minute. If you lose calcium in the extracellular compartment, you can disrupt cardiac muscle cell contraction. Now, there is some calcium stored in the smooth ER that can't be released, so you don't get the second wave of contraction of cardiac muscle cells if you lose the calcium in the extracellular space. And I talked about the cardiac markers like troponin T and troponin I, which can be detected in blood chemistry even up to several weeks after a myocardial infarct. And we talked about atrial natriuretic peptide and brain natriuretic peptide. Brain natriuretic peptide from ventricular myocytes has a function similar to atrial natriuretic peptide. Brain natriuretic peptide, as it slowly increases, suggests that patients might be undergoing congestive heart failure.